So my name is Florian Krammer. I'm a professor here at the Department of Microbiology at the Icahn School of Medicine of Mount Sinai. And if there is no pandemic, my laboratory works on influenza viruses, creating new vaccines against influenza, provide broader protection, and that might also provide protection against new emerging pandemic influenza viruses. We also work on arenaviruses and hantaviruses, which can cause lethal disease. And so we're mostly interested in interactions between antibodies and viral glycoproteins on the surface of viruses. And so in January, we heard about this atypical pneumonia. And actually on January 10th, the genome sequence of the virus was released. And as many researchers worldwide, we were very excited to see that what the sequence is. And it turned out to be a beta coronavirus similar to SARS coronavirus from 2003. And when we saw that, we started to work right away on reagents that would allow us at some point to understand the immune response to this virus. So my lab is specialized in measuring immune responses against these viruses. And so we synthesized genes for the surface glycoproteins of the virus right away. We started to make these proteins recombinantly. And then we started to set up different assays that we could then use to measure immune responses in infected people. And it turned out that these assays were relatively useful. We did that pretty quickly. On March 18th, if I remember correctly, we put a preprint out in the public showing the first serological assays that were able to measure antibody responses from infected individuals. And it became pretty clear pretty quickly that people who have not been infected or not been exposed to SARS coronavirus 2 have absolutely no antibody responses against the surface glycoprotein of the virus. And that also explains a little bit why the virus spread so fast. And so it turned out that it's pretty important to measure these antibody responses. Mount Sinai really led the way in the United States for establishing plasma therapy. As you know, there's not really treatment options for SARS coronavirus 2 currently available. But one of the options that have been used throughout the last 100 years, but also during this COVID-19 epidemic in China, was to use plasma from people who had been recovered and who had neutralizing antibodies, and then use that plasma to treat severely sick patients. Now, what you want to do is you want to find donors that have very high titers of antibodies. And that's where this assay came in pretty handy. So we had a lot of interactions here between the Department of Microbiology, where I am located, where we really focus on virological research, and the pathology department with Dr. Carlos Cordon. And so they set up our assay very quickly in the clinical lab, and we're then able to look for plasma donors and recruit plasma donors. By now they have tested more than 11,000 individuals, and more than 200 people have been treated with convalescent plasma here at Mount Sinai. And so we are continuing our work. Now we're focusing on looking at how stable these antibody titers are in individuals that were infected. We are looking at how neutralizing these antibodies are. And in the long term, we have now started studies to understand how protective these antibodies are. And the goal is to establish a correlate of protection, a titer, that if you have it, would tell you that you are protected from infection with SARS coronavirus 2. That's right now important in case you have been infected and there's a second or a third wave of the pandemic, because you might want to know if you're protected if that wave hits. But it's also very important for informing vaccine development and vaccine work, because if you get vaccinated, you also want to know if the antibody response that the vaccination induced is really protective or not. And so those are the major fields that we are focusing on right now in terms of coronavirus research. And we'll do that going forward. We're planning to conduct several studies for the next 1.5 years. We'll follow the virus. It can be assumed that there will be one or two more waves that are going to hit the population, likely in fall, maybe also in winter. And we want to be prepared for that. And we really want to understand what it takes to be immune to this virus. And so if you're interested in contributing to this kind of research, or research that's going on in our Department of Microbiology in this direction, where a lot of studies are ongoing on COVID-19, you're very welcome to contribute to our work, and we're happy to receive any kind of donations. So thanks a lot, and stay safe.